All right, folks, it's been a little while since I've been with you. The second exam is still up and until midnight on the 31st, which is tomorrow. It's available. There's a second attempt up there now that you can access. If you need to try that again, you can do that. I won't be hopefully doing two attempts on subsequent exams. So this is a chance for you to get back in, work the bugs out on that exam on prokaryotes and eukaryotes. So we're going to start viruses, and this is the first lecture. I want to try to keep this segmented into short 15, 20-minute segments. I don't know how successful I'll be because you know how I am. I really enjoy this stuff. So let's get started on this. I'll pull up the PowerPoint. So here we are, Chapter 6, Viruses, Introduction to Viruses. A timely subject, right? This is something that really, I think people who want to know more about this coronavirus would be chomping at the bit to start to discover and learn more about. So let's jump into this. So viruses, relative to other microbes, have only been known for a little more than, say, 100 years, 120 years, something like that. Back when Pasteur was doing his work with bacteria and working out, the agents of fermentation, and eventually came around to developing the germ theory of disease, that was all focused on bacteria. And occasionally they'd run across a disease whose origins, whose causative agents were unknown. And it was very troubling. But as was often the case with Pasteur, he was kind of sentient and saw into the future and put forward this idea that there's another agent. There are other microbes that we are incapable of detecting, that they were incapable of detecting with technology as it stood at that time. And rabies was such a disease that was troubling for him. So in 1884, he put forward this idea of the virus. And later on, there was confirmation that, in fact, there are viruses. But the first viruses that were discovered and more fully characterized were those that were infecting plants. And a disease known as tobacco mosaic virus was determined to have, a disease called tobacco mosaic was determined to have been caused by a virus. And that was Iwanowski and Bayerink in the 1890s. And then thereafter, as often happens in science, people began accruing more and more information, and a whole field, a whole discipline of biology developed out of this, and we now call it virology. That discipline had its origins early on, but it was officially active in the 1950s. And so what came out of that is, viruses are biological particles. They're not truly living because they don't have cellular structure. Nothing about them really that is even remotely like that which we see in cells, except for the fact that they have genetic material. We'll be talking more about that here as we go. They don't have the phospholipid bilayer membrane, except when they acquire an envelope. And they don't have metabolism. They don't have their own ribosomes. They don't have vacuoles, vesicles, all of those things that we would expect to find in a living cell. They do have definite size, shape, and chemical composition. So one big question that I'm often asked is, how did these viruses get started? Does anybody have any idea about that? And there are people who put forward hypotheses about it, but there's really no definite, sort of consensus on that. Sort of a chicken versus the egg kind of discussion. Which came first, viruses or or living things? Did cells come first or did the viruses come first? My guess is that probably cells came first, and then later we had these rogue particles that developed that began to parasitize them, but we really don't know for sure. They are, in the grand scheme of things, the most abundant, in terms of numbers of particles, the most abundant of all of the microbes on the planet. There are more of them than any other forms that we can think of, just in sheer numbers. So it's just astronomically large, the number of viral particles that are out there. Of course, they're very, very small, and so collectively, the mass that they would make up would be pretty limited. But they're everywhere. They're ubiquitous, and they're dependent on living things for their ability to replicate. And thus, they have played a role in the evolution of all organisms. Viral particles arise and they develop the ability to infect a host cell, and it's going to compromise how the host cell works. So if you have a population of hosts that is variable with regard to how they deal with the virus, that population is going to evolve, probably becoming more resistant to the virus. The virus itself over time should become less virulent, and thus less likely to cause harm to the host, because that's efficacious for the virus. If they aren't killing their host, they're not killing their source of resources for replication. So this is called coevolution, when we have forms evolving in response to one another, sort of in an arms race fashion. So, that's sort of the good news of the virus, like the one we're dealing with now, the SARS coronavirus number 2 that causes COVID-19. As time goes along, it should become less virulent. Of course, the organisms that are being affected by it would also evolve in response to that. And of course, it's going to take a lot longer. For reasons that have to do with generation time and things like that. But anyway, yeah, so viruses are, you can view them as a kind of pressure on organisms that leads to their evolution. So all organisms are products of past infectious agents having affected their populations. And that's an ongoing thing, unfortunately, as long as there's life on this planet. And so this last thing here we've made abundantly clear in the past. Let me try this. And I'm trying to use my pen here, see if it works. Doing this with my finger. Oh, yeah, there we go. Viruses are obligate intracellular parasites. Obligate, of course, means that they have to. It's necessary. It's essential. Intracellular means they go into cells. And parasites means that they take resources and thereby negatively affect the host while they in turn benefit. Okay, so it's a plus for the virus. It's a minus for the parasite. Okay, so here we've got a sort of image which shows the size of different kinds of microbes, give you an idea about how they vary in size. In the background, this big blue cell here is a yeast cell. Sorry, not very clean, but that's a yeast cell. Saccharomyces cerevisiae. It's one of the fungal organisms, so it's a eukaryote, and those cells are very large, relatively speaking. And so that's there to kind of give you, an idea about the top end as far as cell size goes. About 7 micrometers in dimensions from, along the longest length of that cell. And then we've got the bacterial cells over here, E. coli, 2 micrometers, streptococci, quite a bit smaller than a rod, about 1 micrometer, and then the rickettsias, and we could include in here also the chlamydias. They're very small, approaching the size of some viruses, and that's why for some period of time rickettsias and chlamydias were in fact considered to be possible examples of viruses until later it was cellular. And so that's sort of the range of prokaryotic cell size. And then the remainder of the things up here, numbers 1 through 10, are all viral particles. And you can see the ranges of sizes given here, not in micrometers, but in nanometers, so billionths of a meter. All right, so these are small enough that we actually have to take the scale of measurement and drop it down an order of magnitude. So the megaviruses are very large as viruses go, almost a micrometer, and then we just drop down from there. Have a range of viral particle sizes. The virus here, number 5, is the HIV virus, and number 6, the influenza virus. In that range there is about where we would expect to see this SARS coronavirus that we've got sweeping through our population currently. They're similar in a lot of respects to those two, physically and in terms of size. So, and then you can see the sizes go way down from there. Numbers 7 through 10, those guys are all very tiny viral particles. And you'll notice that in comparison to the numbers that precede them, there are no outer coverings with little tiny spikes, except for number 7 has some spike-like structures in it. But numbers 7 through 10 are considered to be naked viruses. They lack an envelope on the outside, and we'll be talking more about what that means. So, but anyway, those are very, very tiny. You see the adenovirus, number 7, which causes some symptoms involving inflammation of mucous membranes, similar to colds, a cold virus. The T2 bacteriophage is a virus that attacks bacteria. It infects bacterial cells. They're not immune to this. They can be infected by viral particles. And then polio is caused by the poliomyelitis virus. You can see its size is very small, 30 nanometers. And then finally, yellow fever, which was vectored by mosquitoes and has had some significance in this region in West Tennessee, going back into the 19th century and possibly before. Yellow fever epidemics would sweep through. It affected Brownsville, it affected Memphis, and led to some horrific consequences. Affected, New Orleans and other places in the Delta region. And then finally, the last thing up there, number 11, is a hemoglobin molecule, one tetrameric protein out of a red blood cell. And so those are very tiny. If you consider red blood cells themselves are kind of small, and packed within them are millions of these hemoglobin molecules. That gives you an idea about the very, very tiny size of some of these viral particles. They approach the size of just a single protein. So viruses tend to be pretty small. That's the upshot of that. And so, like it says here, and as I pointed out previously, they bear no resemblance really to cells. They lack all of the important machinery that would be required for a cell to maintain metabolism. So they can't do any of that on their own, which is why it's so difficult to develop an antiviral drug. You don't have anything to attack, really. You don't have anything to disrupt that's ongoing within a viral particle to shut it down. Right, so that's a bit of a problem. They don't have, especially especially what's deficient is the protein synthesizing machinery. No ribosomes of their own and no enzymes and other types of RNAs and so forth to keep that process going. And down here we've got this little branching diagram which shows the components of a viral particle. The basic things are here. They'll all have a capsid, and they will all have some sort of genetic material. Note, though, that it says here or DNA or RNA. Either DNA or RNA, not both. You won't find those concurrently within the one cell, and that's important. That's helpful in categorizing the different viruses, looking at the type of genetic material that they have. And there's some other things we have to say about that because the story is a little more complicated than saying that they either have DNA or RNA. There's more details that we have to kind of parse there. And then these items here, some of them have an envelope, some of them don't. Okay? So there's some variety there. And then this thing here, proteins, matrix proteins, and enzymes, those may or may not be there as well. So there's some variety among them with regard to those things. So there's a covering and there's a core. And that's about it as far as what we have in viruses. They're very simple, which is amazing, in light of the fact that they can wreak so much havoc in populations as this is doing to our human population right now. So capsids. Capsid is proteinaceous. That means it's made of protein. And it covers the genetic material. Okay? So the most basic viruses will have just a capsid and just some genetic material, and that's it. Okay? And capsid shapes vary. The two capsids that are shown here, this blue one, that's a capsid, is called an icosahedral capsid. Icosahedral capsid, I'm sorry about the quality of my writing with my finger there. It's not really good. ICOSAHEDRAL. It looks like a little origami sort of sculpture to me, a little paper sculpture. Little folded pieces of paper could be fashioned into something like this. So these little triangular plates in here are the facets of the capsid. Those little triangular pieces are called capsomers, right? You can see they're all sort of wedged together to form this really geometrically kind of, definable structure. It's got these little points on it, okay? These little points. So that's a defining feature of an icosahedral capsid. And this virus down here, this one has a green colored capsid in this image. That's its capsid. But note on the outside of that is this purple sort of covering that's got these little spikes in it. The spikes are made of protein, and the purple membrane that they're embedded in is the envelope, which is made of phospholipid. It's a phospholipid bilayer, which it turns out was derived from the host organism within which this virus was created. Right? So they don't make their own envelope. They steal it from the host cell. And if you can imagine what would happen, a cell that's been infected by such a virus that steals membrane as it leaves that cell, that cell eventually is going to be compromised to the extent that it dies. It can't keep up with trying to repair the damage that the virus is causing as it leaves the cell. So yeah, the virus at the top here is a naked virus. It's called a nucleocapsid virus because there's the capsid on the outside and the nucleic acid within it. The enveloped virus is also a nucleocapsid virus, but it's enveloped. It's not naked, right? So anyway, there are those terms that I think it's fairly straightforward, right, to see the difference between a naked versus an enveloped virus. Let me just point this out to you, though, since we're on the subject. The enveloped viruses like this one are typically going to infect only organisms whose cells don't have a cell wall, right? It doesn't mean, you know, that organisms that don't have that organisms that do have cell walls are never infected. It just means that the ones that infect organisms that don't have cell walls are more often than not envelope viruses. And frequently, of course, that means animals, all right? So humans can be infected by these envelope viruses, as can any number of other animals, because we are eukaryotes that don't have cell walls. We can also be infected by these naked nucleocapsid viruses as well. It's just that when those viruses are attacking our cells, they're not going to leave and carry away an envelope with them. It's as simple as that. Okay, so, but when we're talking about envelope viruses, just know that one group that's especially likely to be infected by these are the animals. Okay, so keep that in mind, because we're going to talk about the coronaviruses. Um, they fall into that category. They are enveloped. So here's another type of uh, viral particle. Now, not all capsids are the same. So we have those icosahedral capsids, but this type of capsid here is a helical capsid. You can see the capsimers are these little kind of capsule-like things. They look like little pieces of gum or Tic Tacs or something like that to me. And you can see that they're conformed into a tube, right? They're wrapped into a tube. And so this up here is just showing a little chunk of one of those tubes. And then as it's assembled, it's going to get longer like this, right? It's going to grow into a bigger and bigger tube. And within that will be sort of coiled the nucleic acid and contained inside of that, right? And there are some viruses that, that's how they're structured. They are just the helical capsid and the nucleic acid within them, right? A naked helical type of virus would be the result. Now, some of these guys do in fact acquire an envelope, so they can do that as well. All right, so yeah, so those are the helical forms, and this just reiterates what I said earlier about the icosahedral capsids. Oh, here's a better sort of description of how those are put together. The icosahedron, that sort of geometric shape that these things take, has 20 sides and has 12 corners. So that's one of those memorization things. Just remember that. Icosahedral, 20 sides with 12 corners. And here it shows a variation of this type of capsid. This one has little tiny proteinaceous fibers that hang off, or you can call those spikes, I guess. But those are going to be used by this particular type of virus as structures to attach to a host cell. Very important thing. Viruses somehow or other have to attach to a host cell first, and this is one way that can happen. And you can see that those little fibers there, those little guys, are attached to these little corners of that particular capsid. So that's a naked icosahedral capsid virus that has a naked icosahedral capsid that has attached to it little fibers. And this is an electron micrograph of the same type of virus to show that it really exists. So viral envelopes like it says here, mostly animal viruses. That envelope is acquired, taken from the host cell as it leaves. It's got the phospholipid bilayer membrane all through here, phospholipid all the way around, stolen from the host cell. Okay, it cannot make that. And then these little spikes in here are, in this case, for this particular virus, of two varieties. Some of them are hemagglutinin spikes, which are often just designated with an H, and then the others are neuraminidase spikes, designated with an N. The types of viruses that have H and N spikes are the influenza viruses, and they are named. You see this every year when they come up with a new vaccine for the particular virus that is predicted to be most abundant that year. They will tell you that the virus, that the vaccine is most effective against the H1N5 or H2N7. So there are different H's, right? Hence the different numbers, and there's different types of N's, different types of neuraminidases, and so that's what accounts for the different numbers of those. So whatever combination of H and N spike that the influenza virus has will be indicated by that numbering system, right? I think a few years ago it was H1N5 that was especially problematic, but next year the people who are in charge of keeping tabs on what's abundant and what seems to be reproducing most abundantly, those folks, epidemiologists working for the World Health Organization or the CDC or whomever is in charge of that, they would indicate which influenza virus to watch out for using those designations. So the spikes are used by the virus in obtaining access to a host cell. When this particular virus leaves and goes looking for a new host cell, it's going to use its spikes to gain access to the new host, attach to it, and to make its way on into the host cell to begin the process of infection. And then you can see inside of here, you've got underneath that envelope, you've got matrix protein. That's not something we always expect to have in a virus, but this particular one does. It's just some protein that helps to protect it a little more. And then the capsid is actually inside of here. The nucleocapsid is inside of here. You can see that this particular capsid is helical in form. That's actually not like the flu virus that influenza viruses have. Icosahedral capsid, but this particular form has a helical capsid with the genetic material shown in red here kind of coiled up inside of it. So that's an envelope virus, how one of those might look. So yeah, this just is more information about envelopes and their role in protecting the nucleic acid. The capsid helps out with that, of course, as well. It is the main protector of that. But also the envelope or capsid, depending on which is outermost, will help to bind to a host cell surface and penetrate. All right, so we have viruses that have helical capsids. We have viruses that have viruses that have icosahedral capsids. We have naked viruses. We have envelope viruses. And then we come to this category, the complex viruses, which sort of combine elements of different capsid styles and are considered then to be atypical of viruses that we would expect to find in the eukaryotic organisms, infecting eukaryotic organisms. These infect bacteria, all right? the complex viruses are mainly those that infect bacteria. One exception to that would be the pox viruses, smallpox and cowpox. They're unusual in that they don't have the typical sort of capsid. They don't cleanly fall into the helical or icosahedral capsid categories, but instead have these lipoproteins that are found in sheathing the genetic material. But a bacteriophage or phage, I don't know, I've heard people pronounce it both ways, is a type of virus that infects bacteria. The PHAGE root word there comes from the same root that you see in the word phagocyte. It means to eat. And so they're so named because when they infect bacteria, let's say they infect bacteria growing on an agar plate, they will literally appear to more or less just dissolve those bacteria and eat them away. And you'll wind up with a plate that eventually doesn't have any plaques on it anymore, doesn't have any colonies growing on it because the bacteria phages have affected it. So here at the top of this thing is the capsid, which is icosahedral, and it's got the nucleic acid within it. You see a part here, a sheath, that is a helical coil of protein. So we've got a little bit of icosahedral, a little bit of helical character in those two places. And then you can see these sort of creepy looking spider leg type of things here. These tail fibers are used in helping the virus attach to the bacterial cell. It looks kind of like a little, lunar landing module or something, a spacecraft. So here, let's say this is your bacterium, an E. coli. What this thing would do is attach like this. A finger. But so this is the virus on the outside attached to the bacterium, and it'll have its genetic material in there. What it does then is it literally, this whole thing sort of plunges down, and you can see these tail pins here are sharp. They puncture the bacterium right in there, and literally then this genetic material, the nucleic acid, whatever that is, DNA or RNA, will be injected into the bacterial cell and will become active there in the process of replicating more and more and more of these bacteriophages. So this whole assembly of protein on the outside, the capsid, the sheath, and the tail pins, that stays outside. The only thing that will penetrate ultimately is the genetic material itself. And so that's typical of bacteriophages. And the reason for that is the reason that it has to be that way, it seems, is that the bacterial cell is covered with the peptidoglycan or peptidoglycan and LPS rich cell wall, which makes it really difficult to take these large structures through. But they can be penetrated, and the genetic material can enter. Right? So we'll never see these proteins in this assembly making their way into the cell. We'll only see those after infection is completed and the bacterial cell is ready to, essentially break open and release the viruses. All right, so yeah, this kind of gives you more of an idea of the range of variety in viral particle structure. So up here, this guy is your pox virus, a typical pox virus. And it's described as being a DNA virus because its genetic material is composed of DNA. So there's no icosahedral capsid, there's no helical capsid, there's no envelope. This is strictly just a bunch of lipoprotein, and you can see it looks like little fibers, kind of wrapped continuously around the genetic material, more or less a haphazard fashion. All right, it's kind of like if you were to take a piece of yarn that's very long, and just continually wrap it to form a ball of yarn. That's kind of what you've got with this pox virus. The second one down here is another form of bacteriophage. You can see it's got its sheath, it's got its nucleocapsid, the genetic material inside of that, and down here is the point of attachment. So this particular one doesn't have the little tail fibers like the other one that I showed you previously. So there's some variation. Up here, we've got our envelope viruses. You can see some of them are like this, where there's a helical capsid containing genetic material, a helical nucleocapsid since it's got genetic material in it, and then the envelope on the outside with these little spikes. Here's an envelope viruses with spikes as well, but this one has the icosahedral nucleocapsid. So either of those are possible. And again, these would typically be expected to infect animals. When they infect anything, that's typically who they'll infect. Here's another envelope virus. This is called a rhabdovirus because it's shaped like a bullet. Apparently, the term rhabdo, that root comes from that sort of shape, refers to that shape. And you can see it's got envelope material with spikes. And then inside of here, that coil is the nucleocapsid, which is helical. And then here's another form that has a helical, icosahedral capsid, this guy. And you can see the vertices of the icosahedral have little bits of phospholipid stuck there. So this one is envelope, but only partially so. All right, so the materials that are found there at the vertices of that icosahedron, these little clumps of this golden-looking stuff, that would be your phospholipid and would be positioned out there. So it is technically enveloped as well, although not to the same extent as these others. All right, that one has, it appears to have, it's genetic material, this stuff in here. This is not capsid material in here, that's the genetic material. The capsid is the blue. So that's a tough one to kind of make any sense out of, but that's what. And then you've got your naked forms here, which can be helical or icosahedral. There's no hint of envelope in. All right, so this down here gives you kind of your whole range of possible variations of viral structure. They can be complex. They can be enveloped with a helical nucleocapsid. They can be enveloped with an icosahedral nucleocapsid. They can be naked with a helical capsid or naked with an icosahedral capsid. So there's 1, 2, 3, 4, 5 different variants of form that you can have among the different viruses. Right? They can be complex. They can be enveloped, helical, enveloped icosahedral, naked, helical, naked icosahedral. All right, and these numbered items in here are just examples. There are, of course, many other viruses besides just these. But any one of those would fall into one of these 5 categories, if that makes sense. All right, so let's do this concept check. And then I'm going to go ahead and stop this video and post it, and then I'll make another one that you can digest these things in chunks. All right, so how would you describe this virus? This is an electron micrograph, and I'll just tell you the dark color around it is sort of like shadow. That's really nothing. That's not any part of the virus. What you see here is kind of an elongated sort of stick. Okay? So you don't see anything at all that has a geometry that looks like this, right? A little sort of container with corners on it. There's nothing like that. So that eliminates the possibility of icosahedral virus. All right. So the only other option would be a helical capsid. And likewise, we don't really see any evidence of envelope material either. As I said, this dark stuff out here, that's just a shadow around this thing. So it's lacking anything out there. It'd be naked. And so the uh, the choice here is B. It is helical and all right. So I'm going to go ahead and stop the video there and come back at that. I apologize for not being there in person. I mean, that's nothing we can help. But we'll get through it. All right. So hang in there, and like I say, there's a second attempt available on that exam 2. Want another crack at that? You can do it. Get on there and that. Have until tomorrow at midnight, 11:59 on March 31st. All right. So, all right.